want to welcome everybody to the Neuroscience seminar this week. Our guest is the intrepid Dr. Ari Rosenberg, who, this is the fourth invite that finally got him here for a talk. In the first invite, he was stuck in a snowstorm somewhere in Wisconsin on his way to the airport. And the next attempt was disrupted by COVID virus. And finally, we're getting him here remotely to give a talk to us. I'd like to let Carmen introduce Dr. Rosenberg. So Dr. Varela, take it away.
Thank you, Kate. Thank you, that's great. Hi, everybody. Welcome to the seminar today. As Kate was saying, we have Ari Rosenberg with us today, joining us from the University of Wisconsin in Madison. He is an assistant professor in the Department of Neuroscience. And Ari's lab works in the investigation of the visual system, the mechanisms by which we perceive 3D vision and how the visual information is integrated with the information from other sensory systems. His lab has been in Madison, I believe, only for about 5 years or so, but we are starting to see like a bunch of papers coming out, so they've been very productive. And it's really a pleasure to have him here. They use a number of different techniques, from psychophysics in humans to extracellular recordings in macaque monkeys. They also use computational work, as we are going to hear about. That's what we're going to hear about today. And they have this very interesting preparation in which they can stimulate both the visual system and the vestibular system to try to understand how we build our perception of the visual world as we move through it. And so it's very exciting to have Ari here today. It's also very exciting because not many years ago, Ari was a student in the Honors College. And so he actually started his research career working with several faculty in the psychology department and also in the Honors College. So a real pleasure to have you here, Ari, and I'll leave you all with your talk.
Thank you, Carmen, for the introduction, and thanks everybody for coming to the virtual seminar. I really like to give interactive seminars where the audience interrupts with questions or speculations even. So there's a mechanism which I think I can see your hand go up. So raise your hand if you have a question. Don't feel like you cannot interrupt in the middle. If I don't answer, maybe just unmute and say, hey, I've got a question. Because the first few questions might go kind of clunky as I learn to work the interface. So please don't feel like you can't interrupt. It's really a pleasure to be here. I wish I could be there in person. I had a really wonderful day chatting with several faculty who were inspirations to me as an undergraduate, first at the main campus in Boca and then at the Honors College. You know, several of these faculty, Robin Ballacher, Kevin Lanning, Julie Earls, you know, they were really the inspirations behind much of my early science and have definitely shaped my path forward. So it was really a wonderful experience to get to see them again from the perspective of a colleague, and then to meet with some new colleagues like Alon. So again, feel free to interrupt and we'll get started. Today, I will tell you, we'll discuss with you some of my autism work with adolescents with autism and some of the computational work that we've done. This is a pleasure for me to talk about in particular because usually I'm giving talks on neurophysiological studies with macaque monkeys. And so this has in some ways been a side project for me that has been very fruitful and productive. And so it's really exciting to be able to finally be on the road and share some of these results. As a starting point, I think it's worth stepping back and just asking, what is autism? Well, broadly speaking, it's a cluster of neurodevelopmental disorders with a high degree of behavioral heterogeneity. There's some commonalities. There's impaired social communication, such as making little to no eye contact, or misinterpretations or inaccurate interpretations of body language, repetitive behaviors and restricted interests, so rocking back and forth, or an encyclopedic knowledge of dinosaurs or something like Star Wars. Motor difficulties, So unusual gait, walking on the toes, fine motor deficits. In fact, some of the earliest biomarkers of autism are motor deficits. But there's so many other aspects of autism spectrum disorders, or ASD, which can impact individuals differentially. So speech onset delays, for example. Some individuals have a speech onset delay, some do not. Within individuals with a speech onset delay, there is a great deal of variability in how long that delay is. It can go from months to years. Hypersensitivity to sound. Again, some individuals have hypersensitivities, others have hyposensitivities. And there are a number of perceptual consequences in others. But it's somewhere in this intersection of all of these different behavioral observations that autism spectrum disorder, or ASD, is ultimately defined. And if you go into the literature, what you will find is that there's a fair amount of research on causes, genetic, environmental, physiological changes associated with autism, and its behavioral consequences on multiple levels. So we know that multiple genes are generally involved in autism. So idiopathic autism, that is multi-gene mutations, account for about 90 to 95% of ASD cases. Idiopathic autism, single gene mutations are quite rare and probably distinct from what we typically would call autism. We know that there are a number of environmental contributors, air pollutants, in particular heavy metals. Epigenetic factors almost certainly play a strong role. One, as a young father, that strikes me as particularly intriguing is the paternal age. So there's a large jump as the father passes 40 in the probability of a child having autism. Physiologically, we know autism disrupts synaptic function, and this gives us some clue that at some level, at least, autism spectrum disorders must be disorders of neural communication, and that's a point that we'll return to at multiple points through my seminar today. And in terms of the behavior, autism is diagnosed with the Autism Diagnostic Observation Schedule, or ADOS. So it's purely a behavioral diagnosis, which I find particularly interesting given the fact that there's so much heterogeneity in this disorder and how it manifests behaviorally. So we know a fair amount about causes, we know about the consequences, but something that strikes me as particularly missing is the bridge between physiology and behavior. So how do we go from these genetic, epigenetic, physiological changes to these sorts of behavioral consequences? And from my perspective as a computational neuroscientist, is that what bridges these are neural circuits and the computations that they perform. And a handful of years ago now, I made the proposal that if we approach autism as from the perspective of a disorder of neural computation, then we can form testable hypotheses that should make predictions both about the physiological changes and the behavioral changes. And what I would hope would come from that is the ability to intervene in some way, either physiologically or through behavioral therapies, in ways to accommodate the sort of disruptions that might be happening in neural computation to improve, for example, the independent living skills of individuals with autism. A number of years ago, there was a relatively influential review paper from a number of prominent autism researchers who said, quote, "We argue that there will be no single genetic or cognitive cause for the diverse symptoms defining autism." And at some level, I certainly agree with this, but what I have proposed is that we can potentially reformulate autism and how we conceptualize it in a way that would allow for a first principle understanding of the disorder through disruptions in neural computation. And so in my seminar today, the first half, I will focus on the possibility of creating a first principle account of autism spectrum disorder. In the process, I'll introduce the idea of canonical neural computations. Broadly speaking, I just mean computations that occur throughout the brain. And I will introduce the divisive normalization of hypothesis of autism, which we formulated in a publication in 2015. So I'll introduce you to that network model, using modeling as a proof of concept for how a computational perspective on autism might provide new insights. And in the second half of the talk, I'll introduce a behavioral test that we've started here in Wisconsin, which is testing a key prediction of the hypothesis, and show how this reveals the existence of two distinct ASD subgroups, which has implications for personalized therapeutic and educational approaches. And that's going to be ongoing work that's not yet published. So let's first look at neural computations, canonical neural computations. And so as an example, let's consider linear filtering. So perhaps you're familiar with the receptive field models of primary visual cortex. These have their history in the work of Hubel and Wiesel, who discovered orientation selectivity in primary visual cortex, which was one of the principal findings leading to their Nobel Prize. What we know about V1 neurons, or primary visual cortex neurons, is that they have these elongated receptive fields, and they respond to the presentation of light increments or decrements. So they'll have subdomains which will be which, if you shine bright light in an oriented way, will elicit positive responses, facilitate activity. They also have subdomains that are elongated and looking for decrements in light. And so presenting a dark bar here will elicit a positive response. And if you, because they act like linear filters, if you present a dark bar over one of those domains that is looking for a light increment, you'll get a negative response. And so this is a linear filter model. It's a canonical computation. So linear filtering is something that you find throughout the brain, both in sensory and motor systems. And it has a very strong prediction. It's just that the weighted input of so the product and summation of the visual stimulus and the receptive field of the neuron is exactly identically equated to the response. And so here's the response on the y-axis, the weighted input on the x-axis, it's just a line. And so some canonical computations like linear filtering exist at the single neuron level. There are other computations that are canonical at the single neuron level, which are very important, in particular rectification. So neurons communicate through spiking events. They fire action potentials to convey information. And so there's no negative responses. And so there's a rectification that turns all those negative responses into an output of 0. And so rectification, or this spiking nonlinearities, is another canonical computation, something that you see from effectively all neurons in the brain. But these are at single neuron computations. Not all computations occur at the single neuron level. Many computations occur at the network level. So let's look at how that might look. So you can imagine a sensory stimulus. So for example, the sensory input that's projected onto the retina of one of your eyes, which is fed forward in some way to a population of V1 neurons. Some prefer vertical orientation, some horizontal orientations, others prefer oblique orientations. And in a very simple model of what the visual system is doing, the convolution or product of that receptive field and whatever that arbitrary sensory stimulus is gives rise to an output, and in some feed-forward way that that population activity can be used to recompute whatever the image was. Now, we know that this is a very simplified model because it only contains excitation, and this can't possibly be what the brain is doing for two reasons. One, if the brain was purely excitatory, we would be in epileptic states all the time, because we would reach some threshold where all of the neurons in all of the brain are excited. And so And the other is that we know that the brain cannot be purely feed forward and linear, because this kind of linear feed forward processing doesn't allow for the possibility of the creation of new kinds of information. And information creation is something that's that all humans and living organisms do. So there must be some form in particular of inhibition that's built in. And one way that you can build that inhibition in is through inhibitory interneurons that can do something like sum the population activity. So it's not just a purely feed-forward model. All of these neurons, excitatory neurons, are providing input to some inhibitory neuron. And that neuron in turn provides inhibition on each of those excitatory neurons. And inhibition can take on a couple of forms. It could be subtractive, it could be divisive, so division. Either way, you will quench activity in a way that can prevent, for instance, epileptic states. And one particular form of inhibition that seems to be particularly important within the brain is divisive normalization. In a feed-forward with lateral connectivity network with this kind of inhibition, you can write down the response of any one of these neurons as the ratio of that excitatory output, that linear filtering, divided by some function, which in its simplest form is just a parameter that prevents instabilities in the network, plus some gain term, which scales the amount of inhibition that's impacting the activity of each of the neurons. And so in some sense, divisive normalization controls the ratio of excitation to inhibition within the network. So how does the EI, or excitation to inhibition ratio, affect neural responses? Here's an equation, that equation describing the activity of one of the neurons in this kind of network. On the x-axis of this plot, we have the excitatory input. So how strong is the visual input against how strong is the neural response. And as a consequence of divisive normalization, the activity doesn't grow boundless, it saturates at some level. And if we focus on this parameter C here, which you can think of as a volume knob on the level of excitation to inhibition, then if we decrease C, we increase the EI ratio, because then as you decrease C, the numerator gets smaller. And what that does to the neural responses is increase the level which the responses saturate. So with less inhibition, the responses saturate at a higher overall level of activity than if the inhibition is stronger. If we instead changed this stability parameter, what you would get is, rather than a function that looks kind of like a logarithm here, you get a S-shaped curve. So it pulls the inflection point to the right or to the left. Okay, so this, when I discuss canonical computations, these are the kinds of computations that I'm referring to. So how might this be related to autism? If you go into the literature, both with human studies and with animal murine or rodent models of autism, you see that there's a fair amount of evidence suggesting that autism is linked to an increased EI ratio. And so in 2015, we proposed that you can define a spectrum of EI ratio with this kind of network model, and at some typically developing version of this model, the inhibition component of that is tuned in a way that matches what we find in typical development. And as you fade out that inhibition, you reduce the amount of communication from that inhibitory network, you increase the EI ratio, and you potentially could give rise to autism-like behaviors. So autism reflects a weaker divisive normalization signal in our original hypothesis. And this is consistent with, for example, the high comorbidity of autism and epilepsy. one thing that you might notice is that by fading out this inhibitory component of the network, the neurons, the excitatory neurons that are doing these computations are more like islands. They're more feed forward, less lateral connectivity. So in some sense, that inhibition is important for telling, for example, this neuron what this other neuron and its other neighbors are doing. And when that inhibitory circuit is attenuated, the excitatory coding neurons have less information about what the others are doing. And so in some sense, what we proposed is that ASD reflects less social neural circuits, at least in this model. And so one thing I would like to emphasize is that this is likely a simplistic perspective on what autism is, in the sense that an attenuation of divisive normalization signals. There's almost certainly much more going on, but this provided a foundation for creating systematic predictions that we could then go and test. And that was our starting point. And as I'll show you in the second half of the talk, we've had to extend this model in some rather exciting ways to account for some novel data. Again, if there are any questions, please interrupt. Okay, so how can the divisive normalization hypothesis of autism make predictions that match known behavioral findings? This was a question that we wanted to first look at. So we built a neural network model and used that neural network model to see if we can account for known behavioral findings in the visual psychophysics literature associated with autism. And one of the papers we picked up on was from Robinson and colleagues in 2013, who measured the spotlight of attention in autism. So there's a well-known finding that presenting an attentional cue near a target will improve detection. And so in this study, they presented circles with an opening either at the bottom or at the top. And the participants' job was simply to report top open or bottom open. So in the task, they had participants fixate at a cross, and this participant had to always fixate at this cross. And then they would flash an attentional cue and then a target at variable distances from that cue. So in some trials, the cue and the target were right on top of each other or very close. And in other cases, the cue and the target were very far apart. And when they looked at the behavior of the subjects, then here TD, reflecting typically developing, showed a well-known pattern. Your performance is best at target distances that are very close to the cue, and worse at target distances far from the cue. And when they repeated the study with age and IQ matched participants with autism spectrum disorder, what they found was that there was a steeper gradient in the fall off of that attentional cue. And so the authors, interpreted the result as a sharper gradient of attention in autism. And this didn't seem to just reflect group differences, but individual differences. So you could calculate that performance gradient, so how steep this fall off is, both for the group or for individuals. And you can plot that as a function of some measure of autism symptomatology severity. In this case, they used AQ, autism quotient, which is a simple metric from the Baron-Cohen lab. And so larger values reflect greater autism severity. And what they found, and one nice thing about this measure is you can calculate it for both typically developing and ASD subjects. And what they found was that there is a strong relationship between autism symptomatology and the steepness of this attentional gradient. And so we wanted to determine if we could account for this sharper gradient of attention in autism through a computational model of attenuated divisive normalization. Okay, so the take home from this data was that more severe autism symptoms reflect a narrower field of attention or a narrower spotlight of attention. So how did we go about modeling this? We developed a neural network model of primary visual cortex. So this was a relatively straightforward model. Different units or model neurons were selected for orientation and position, just as they are in primary visual cortex. We implemented lateral connectivity through divisive normalization. So we have that equation that I showed you before. This feed-forward excitatory component divided by the divisive normalization signal. And we added one layer of complexity, which was a top-down attentional gain signal. And this is incorporated here, where it's a function, the attentional signal is a function of distance. So you get a stronger gain when the target is close to the cue, and a weaker gain when that target and cue are further apart. So this is just a Gaussian function centered on the cue's location. Okay, and so what we will then do is decrease C to simulate autism. And I'll point out that the only thing we're changing in this model is in the divisive normalization component. We're not changing anything about the top-down attentional signal. And then we'll use the gain of the population activity as a proxy for behavioral data. So I'll give you in the next slide what I hope is an intuition for how we can use population gain or the amplitude of the population response as a proxy for behavioral performance. And this follows from work of Weijie Ma and other theoretical neuroscientists from the mid-2000s, and some applications that we came up with later for multisensory cue integration. So in this case, we have the preferred stimulus orientation of each neuron in the neural network. So neurons which prefer an orientation of 0 prefer horizontal bars. Neurons that prefer 90 degrees prefer vertical bars like this one. And on the y-axis, we have the response of each neuron in the population. So you present a visual stimulus, a vertical bar, and you look at the activity of each neuron and in the population, and you rank order them according to their preferred orientation. So here's a bright vertical bar. And so you get a very strong response from the population that peaks at 90 degrees, coinciding with neurons that prefer vertical bars will have the strongest response to that vertical bar. Neurons that prefer horizontal bars will have the weakest responses. And we can also present a weaker stimulus. So we just turn down the contrast. So now that bar is not bright, but kind of faded out. And what you would expect is a peak at the same spot, but of a weaker amplitude. And then through a relatively straightforward application of Bayes' theorem, you can reinterpret that population response from the neurons as a probability density function. So now the stimulus orientation, the possible stimulus orientations that might be out there in the world against the stimulus probability. So the most likely stimulus given that population response is 90 degrees, and an oblique bar is very, very unlikely, and a horizontal bar would be even less likely. And so what you should take away from this is that these strong population responses give rise to tall and narrow probability density functions. So there's greater certainty about what the stimulus was when the stimulus response of the neurons was stronger, and the density function is shorter and broader, so there's less certainty when the population response is weaker. So the gain of the population, how tall this response amplitude is directly related to the level of performance. Greater population amplitude, taller, narrower probability density functions, more accurate performance. Is that okay for everybody? Is that clear? In particular for the students?
Yes.
Great. Okay. So we can use the gain of this population activity as a proxy for behavioral performance. So can divisive normalization account for this pattern of behavioral data that Robinson found? Here we have the neural network model, target distance from Q, performance gain, population gain on the y-axis. We can look at the population gain for the typically developing version of the model. And sure enough, you see that the gain falls off as a function of target distance from Q. It then does this partial reversal. It never gets quite as high as it does when the target and the Q are close together, but there's this partial reversal. This is a natural consequence of divisive normalization because of spatial scale constants that are built into into the divisive normalization signal. You don't see that in this data set, and it's likely just because they didn't measure far enough out. You see here we've measured out to greater distances, and if you go into the attentional spotlight literature, what you find is in fact that the attentional spotlight isn't just a spotlight, but a difference of Gaussians or a Mexican hat shape. And to my knowledge, this model is the first time that anybody's provided a computational explanation for why the attentional spotlight isn't just brightness, a bright region where you can attend better, but actually has a trough and then some rebound. And so we then turn down C to create the ASD version of the model. And indeed, you find that the gradient is steeper in the autism version of the model. Again, we've only changed C. This isn't a change in any way of the top-down kind of attentional spotlight signal. So can this account for individual differences in the attentional spotlight? So here we plot the percentage decrease in C as a function of the population gain gradient. And you see that as you go from the typically developing version of our network to the ASD version of our network here in cyan, as you move from one to the other or even past that, as you decrease C, you get a sharper and sharper gradient of attention. So what this tells us is that, at least in this case, we can account for both group differences and individual differences in the gradient of the attentional spotlight through changes in the strength of divisive normalization, which is consistent with these findings between group differences between typical development and autism spectrum disorder. Okay, so that's just one example. In the original paper, we provided a number of observations and neural network simulations showing that we can account for a breadth of findings in the autism spectrum disorder literature as a consequence of attenuated divisive normalization. So there's behavioral evidence related to feature complexity. So often you find in the literature that individuals with autism have difficulty processing complex features such as faces, but they have improved processing, better processing of simple stimuli than individuals with typical development. And this attenuated divisive normalization model can account for both of these findings. In particular, for simple features like oriented edges, attenuated divisive normalization creates stronger gains in the representation of simple features like edges, as we saw. But divisive normalization is very important for doing contrast normalization. So there's a lighting source in this room that makes the bridge of this nose brighter than the right side of the nose. And if you're a computer vision scientist, what you would be plagued by is that this bright edge here and this dark edge here creates conflicts in how you would want to piece the components together to create a face construct. And to resolve those kinds of conflicts, you need divisive normalization to do local contrast normalization. So if you have attenuated divisive normalization, you will have problems doing stitching the features together to get complex feature representations. The model can also account for differences in the use of prior experience to interpret current sensory information. One way we demonstrated that was with the oblique effect. So humans and monkeys and octopi and just about everything in the world is more sensitive to vertical and vertical and horizontal edges, and less sensitive to oblique edges. And this is related to the natural scene statistics. So there's lots of vertical edges and horizontal edges. There's the horizon is a horizontal edge. Ground planes have lots of horizontal edges. Trees and buildings are vertical. There's not a whole lot in the world going on that's oblique. And so the visual system is developed in a way that makes you more sensitive to the more likely stimuli, less sensitive to those less likely stimuli. And if you have attenuated divisive normalization, you will attenuate this oblique effect. And you see, in fact, an attenuation of the oblique effect in autism spectrum disorder. Rationality. So in value-based decision-making, humans are incredibly irrational. And in particular, for example, you're presented with three options, two very appealing options and one kind of distractor low value option. That low value option actually will shape how you interpret the value of the higher value objects. That is kind of a ubiquitous finding, particularly in undergraduates. And it turns out that individuals with autism show more rational decision making. And this falls naturally out of the attenuated divisive normalization, because as it turns out, that irrationality that you see in human behavior is well explained by divisive normalization and contextual relationships that are only possible because of the lateral connectivity in neural circuits that's implemented through divisive normalization. Okay, multisensory integration. This is actually one really interesting observation in my mind. So ASD, it seems largely shows no impairments in multisensory or unisensory cue integration. So how you integrate, for example, visual and vestibular information are two different kinds of visual cues. And divisive normalization is known to be this kind of ubiquitous computation that happens throughout the brain. But it turns out the one place that's well documented to have no, for divisive normalization to kind of cancel out, to give rise to just linear summation of neuronal responses, is cue integration. So if you look at this behavioral evidence as a whole, you kind of have this five-card stud relationship between our model and autism. Some places we predict that there should be no change, and you see no change. Some places you predict that individuals with autism should perform better than individuals with typical development, and that's the case. In other cases, there should be deficits in processing, such as in complex feature processing, and that's what you find. So this is all very intriguing and suggests that there might be something to this divisive normalization hypothesis, in particular in attenuation. It can also predict physiological evidence, including greater stimulus-driven activity and larger response variance. This is something that you see in rodent models of autism and is predicted by our model. And also decreased efficiency of information transmission between brain regions. So can this potentially give rise to a first principles account of autism? I think that's still a relatively simplistic perspective, but there seems to be some support for this. So we can account for qualitative differences between individuals with autism. So different brain areas might be affected in autism, giving rise, for example, to individual differences in visual or auditory disruptions. And this seems to be associated closely with divisive normalization. So this parameter C that tunes the strength of the E to I ratio differs across different pathways. This parameter C in the visual pathway is mediated by GABA A. In the auditory pathway, it's mediated by GABA B. So you can imagine a situation where disruptions of just one of these pathways or the other can give rise to different qualitative differences across individuals with autism. And you might also find that this can account for quantitative differences. So how much divisive normalization is affected in a specific brain area. So how much is one of these pathways disrupted? Well, that might reflect multi-gene or epigenetic factors. Okay, so that's the first part of the seminar. In the second part, I'll take you through a behavioral test of one of these key predictions.